Good morning. My name is Alexis Hankerson, and I'm going to discuss the movement of macromolecules. So the first one that I'm going to discuss is estradiol, which is a lipid steroid hormone synthesized in the granulous cells of the ovaries, where it binds to sex hormone binding globulin. The hormone diffuses across cell membranes, modulating potassium and calcium currents through the cystic AMP and protein kinesis A, also known as PKA, signaling pathways, and operates via the G alpha S and G alpha Q coupling mechanisms. It's produced in the endoplasmic reticulum and is generated from estrone by the enzyme 17-beta hydroxide steroid dehydrogenases and can also be synthesized from testosterone. Estrogen plays a critical role in regulating DNA transcription, influencing the menstrual cycle, breast and ovarian function, and brain activity, particularly affecting genes that lack estrogen responsive elements. Estradiol crosses the cell membrane to bind with intracellular receptors, which subsequently regulates gene transcription for protein synthesis. The estradiol receptors complex then translocates to the nucleus, where it modulates gene transcription. Estradiol binds to estrogen receptors within the cytoplasm, leading the receptor complex to enter the nucleus and attach to DNA, thereby regulating gene transcription. This process produces messenger RNA, which then interacts with ribosomes to synthesize proteins that manifest the effects of estradiol on the cell. The next one I am going to talk about is glucagon, which is a protein hormone produced by the pancreas. It's critical in regulating blood glucose levels by increasing and preventing hypoglycemia. It is synthesized in the endoplasmic reticulum, processed in the Golgi apparatus, and stored in secretory granules. When released into the bloodstream via the exocytosis, glucagon is primarily metabolized by the liver and kidney. The hormone binds to receptors mainly in the liver, as well as the kidneys, heart, adrenal glands, adipose tissues, gastrointestinal tract, and pancreas. This binding activates a G protein that stimulates adrenal cyclysis, leading to the production of cyclic estrogen monophosphate, also known as CAMP. Elevated CAMP levels activate protein kinesis, which translates to nucleus and activates transcription factors like CAMP response elements, which is also known as CREB, to promote gene expression. Additionally, the glucagon activates phospholipase C, also known as PLC, increasing the IP3 and mobilizing calcium ions to enhance gene expression. As depicted in the flow sheet, a gene encoding glucagon resides in the DNA of the nucleus. Transcription factors initiate the transcription of gene into messenger RNA, which is processed and transported to the cytoplasm. Here, ribosomes on the rough endoplasmic reticulum translates the mRNA into polypeptide chain that folds into a functional structure in the ER lumen with possible enzymatic modification. The protein is then packed into a transport vesicle that buds from the ER and fuses with the cis Golgi apparatus for further modification. Finally, it is sorted and packed into a secretory vesicle at the transphase, which moves to the plasma membrane and releases glucagon into the extracellular space through exocytosis. And the final protein that I'm going to talk about is the microtubules, which is an intracellular protein found within cells that contribute to structural integrity and organization. They serve as tracks for the movement of materials and help position organelles, ultimately influencing cell shape and behavior. It's composed of alpha and beta tubulin. Microtubules can alter their length to carry out various functions. During cell division, centrosomes anchor microtubules, while basal bodies provide support for cilia and flagella, facilitating cell movement. Microtubules play a critical role in transporting vesicles and organelles, separating chromosomes during mitosis, and moving cellula and flagella. They extend from centrosome to the cell membrane, helping to organize the cytoplasm and position organelles. They are composed of tubulin protein units that pair as dimers, stack into protofilaments, and ultimately form hollow tubes. Each tubulin subunit binds to GTP, which is essential for microtubule formation through a process called nucleation. As GTP converts to GDP, the stability of the microtubule diminishes, potentially leading to its dispensability. Microtubules extend distinct polarity with the beta subunits of the dimer connecting to the alpha subunit of this next. This arrangement results in two ends designated as negative and positive signs. Although they can elongate at both ends, the growth rate is significantly faster at the positive end. And that concludes my presentation. These are my references. This is one page and the second page. Thank you so much.